Our lab focuses on understanding muscle development, degeneration, so when muscle doesn't work properly, and regeneration, so when it gets fixed, in critical severe neuromuscular conditions, diseases of childhood. We focus on using different types of human stem cells, some of which we have also helped to develop and discover and use in the lab, including human so-called pluripotent stem cells, particularly human iPS cells, induced pluripotent stem cells, which are made by giving a special cocktail of molecules to, for example, skin cells, and turn them into super stem cells that we can then differentiate into any cell type that we want with the right recipe. And our contribution to the field and our specialty has been to make muscle out of those, and now we can even make mini muscles out of those. We and others have seen a lot of promising treatments that have worked well in mice, for example, of mouse models of muscle diseases that didn't really work as good as they were working in an animal model when they went to clinical studies. And this is a challenge that is still there very much in not only in our field but in many other fields of life sciences, and we hope that with the sort of strategies that we are developing now to make this research more humanized, in the sense that it's more based on human models rather than doing something in a different species and then try to see whether it works in human, we hope that that will help overcoming some of those challenges. We have several exciting projects in the lab, but perhaps one of the most exciting we have at the moment is a large project which is called MAGIC, and this is we call a consortium, so a team of scientists from all over the world, which I have the pleasure to lead, and it focuses on developing new models of muscle disorders using human stem cells and we say tissue engineering strategies, so that we can then use those models to develop and test gene therapies for muscle disorders. So therapies based on engineering, of tweaking viruses to make them unharmful, and use those viruses to transfer a healthy copy of a gene to muscle in this case, and we hope that this could speed up the development of treatments for incurable muscle disorders.